talking about problems in meiosis, and sometimes you have changes in chromosome structure, and it's not an entire chromosome that's gained or lost. So there can be a breakage of a chromosome, and that can lead to 4 types of changes in chromosome structure. So a deletion occurs when a chromosome fragment is lost during cell division. So here's an example of a deletion. You have genes or segments A, B, C. This wouldn't be a gene, it would be a segment. A, B, C, D, E, F, G, H. And then you go, you lose the D part, and it goes A, B, C, E. And so there's a whole section of a chromosome that's lost, which would mean a whole ton of genes that are lost. So this chromosome will be missing certain genes. Another example is a duplication. This occurs when a fragment becomes attached as an extra segment to a sister chromatid. So a deletion in one will become a duplication in the other often. So A, B, C, D, E, F, G, H. B, C is copied and maybe taken from the other chromosome. So you'll have A, B, C, B, C, D, E, F. So anyway, that's a duplication, meaning you have extra. Another example is an inversion. You'd have all your DNA, so you think it would be fine, but it's not. So you have, the order is supposed to be A, B, C, D, E, and in this case, they're just backwards. They came off here and reattached backwards, so it goes A, D, C, E, C, B, E. So that occurs when a chromosomal fragment reattaches to the original chromosome, but in the reverse orientation. And you would think this kid would be just fine, the kid produced from this DNA, but they're not. The order of the genes actually matters, and that has to do with regulation of the genes, most likely. Another example is a translocation. So a chromosomal fragment joins a non-homologous chromosome. So here's a chromosome, and maybe this codes for feet width or something, and that codes for hair color, and that codes for eye color or whatever. This chromosome has no genes for that at all. It's a totally different chromosome, but this section gets swapped with this section. So they're swapping, but they're not homologous. So MNO is supposed to be over here, and it's not. It's on the wrong chromosome. And so that can cause problems too. So some translocations are reciprocal and others are not, meaning you can swap them between one and another, and sometimes you just take a piece off of one and stick it on the other. What did I want to do with this? Stop here and add this. Oh, so I have to add some more stuff. Okay. So some more stuff. Extranuclear genes exhibit a non-Mendelian pattern of inheritance. This is kind of cool. So not all your eukaryotic cells' genes are located in the nucleus, right? Some of them in the mitochondria. So extranuclear, meaning genes outside of the nucleus. Extranuclear genes are found in small circles of DNA in mitochondria and chloroplasts. These organelles reproduce themselves, and their cytoplasmic genes do not display Mendelian inheritance. That means that you don't get some from mom and dad and have them do the crossing over, blah, blah, blah. You get them all from your mom. So they're not distributed to offspring the same way as all the DNA in the nucleus is from meiosis. So here's an example. The coloration of some plants is determined by the maternal parent only. So the genes for coloration pattern are due to genes in the plastids, which are inherited only via the egg, not from the pollen. Whoops. Because the zygote inherits all of its mitochondria only from the egg or the ovum, all mitochondrial genes in mammals demonstrate maternal inheritance. Several rare human disorders are produced by mutations to mitochondrial DNA. These primarily impact ATP supply by producing defects in the electron transport chain, or ATP synthase. Tissues that require high energy supplies, for example, the nervous system and the muscles, may suffer energy deprivation from these defects. Other mitochondrial mutations may contribute to diabetes or heart disease or other diseases. And that is the end of my story.